Let's get started then with our first speaker. It's my pleasure to introduce to you Chris Chen. Chris Chen is our William Fairfield Warren Professor at Boston University. He's the founding director of our BDC, Biological Design Center. He's a remarkable investigator. He'll tell us about some of his innovations and impact. And I could tell you on a personal level, he's just remarkably collaborative. He brings together both campuses of Boston University with incredible collaborations across divisions, departments, schools, and of course, campuses. So without further ado, welcome, Dr. Chen.
Can you guys hear me? This works? Great. Maybe as the slides are going up, I'll just say that I have no AI material in my talk today. The AI that I use is, hey, what should I tell my daughter when we need to do something about finding summer jobs? And that's about it. So what I'm going to do today is talk to you about some of the work that my group is doing. Oh, I can click. I have control. Okay, great. So what I'm going to do today is talk to you a little bit about some of the work that we do on engineering tissues. And I'm going to try to explain to you why we think that this is so important. And what I'm going to do is just start with this one example, although I think you can all relate to how this is relevant to practically every disease that we can think of. In particular, I'm interested in heart disease. It's obviously very important and leading cause of death, et cetera. Many dollars in health dollars that are lost through the care and taking care of these patients. But one of the things that I want to think about is how do we get to a point where these diseases are not having such a big impact? And one of the things we want to think about is the fact that many drugs that are currently used and developed actually get pulled from the market after they've been approved. 32% end up with a safety signal after they're already in the market. And these are not just cardiac drugs, these are drugs across the industry. And you might ask yourself, why is that? Why is it that we have this massive inefficiency in developing, as you guys heard earlier, spending billions of dollars on a drug, and at the end, oh, causes problems. I guess we shouldn't use it. That is a huge cost of effort, resources, time, et cetera. And what it comes down to is, how do we understand disease and how do we figure out how to develop cures for those diseases? And what I would argue is that most of what we understand about heart disease comes from animal work. And what we all know today is that heart disease in animals, for the most part, doesn't replicate in humans. The severity of the disease, the time course of the disease, the molecular mechanisms that are causing disease are just different. The ion channels that are expressed are different. And so as a result, you might expect that interventions that work in animals end up failing in humans. You can also imagine off-target toxicities if we're looking for, let's say, not a cardiac treatment, but something else, Alzheimer's treatment, that the likelihood that we would discover a cardiac toxicity in those animals is also relatively unpredictable. So how do we get around that? Well, what many people have thought is that the way to get around that is to develop human models. And one way that we study that is in human cells. So the technology to culture human cells has been around for about 120 years. The technology for that has not changed largely over the course of those 100 years. The very first cultures were just taking explants out, putting them into a dish, letting cells crawl out, feeding them with serum. Guess what? We still sort of do that today. But there was a couple of events that changed that course. One is the development of recombinant proteins over the course of the 60s, 70s, and 80s that allowed us to develop more specialized culture medias. And then the development of iPS cells. Many different types of cells that we couldn't culture before, now we're culturable. But what I would argue is that even then, when you take those cells and you culture them on a dish, many of those cells don't behave like the way they do in your body. Same thing. Gene expression patterns are different. That means the target protein is different, the circuitry controls are different, et cetera. So we, again, necessarily don't have a model that predicts what happens in humans. What I believe is that part of the reason why we lose that phenotype is because the cells are not surrounded by the cues that they would normally see while they are in your body, whether it's the physical cues, the paracrine factors, et cetera. And so one of the things that we're interested in is, can we take those cells and then sort of rebuild a tissue around those cells so that they start to behave like they are in your body? And therefore, can we create models that actually are more predictive? If we could do that, I think there are a couple of applications. One is obviously those models then become models for healthy or diseased tissues that we can then use as a surrogate instead of animals or in addition to. And then the other is eventually, if we get really good at understanding those cells, we know what they will do when we use them therapeutically in the body. So those are the sort of ultimate goals. And what I want to do today is just show you a couple of examples of some of the work that we're doing in this space. One of the things that we're really interested in is modeling cardiac muscle. So people have been able to take iPS cells, differentiate them into cardiomyocytes for some time now. And one of the things that we were interested in doing is taking those cells and assembling sort of a muscle-like tissue. And shown here is an example of one of the devices that we developed, where essentially it's just sort of an array of small wells. And then what we can do is we can spin these cardiac iPS cells, iPS cardiomyocytes into these wells. We do that with matrix. And then what happens is the cells grab onto the matrix polymerizes, and then it starts to compact the material as it does in all cell types with all types of extracellular matrices. What we did was we actually put into those wells these two pillars you can see on the left and right within that well. And then what happens is as the gel compacts, the gel kind of gets caught on those pillars. And you notice these pillars are not just sort of vertical pillars, but they have little knobs at the end. And what that does is it captures the gel. And when that happens, then the cells and the matrix align as this whole thing is sort of contracting, aligns along the tips of the pillars to form a bridge, and ultimately a tissue that looks a little bit like an aligned muscle. So we got really interested in using this as a model to model cardiac tissue. And we ended up getting connected with a good friend and colleague now, Cricket Seidman, who's a pediatric cardiologist over at Children's and the Brigham, and her husband, John Seidman, who's a leading human geneticist. And what they've been doing over the last 50 years is basically taking care of patients who have familial forms, hereditary forms of cardiomyopathy, and they've been taking blood from them. And as sequencing technology became available, they started sequencing all of these kids to understand what mutations might be associated with various forms of cardiomyopathy that they were seeing in these patients. They identified many of these proteins themselves, and many of them fall into various categories of cytoskeletal and sarcomeric proteins that are involved in driving the contractility of these cardiomyocytes. One of the things that they weren't able to do is study whether those mutations actually, what do they do in the cardiomyocytes that cause these problems. And so when iPS technology came around, they got super excited. They had this postdoc that spent 7 years taking that technology, generating iPS cells from all of these patient banks that they had, and then turning them into cardiomyocytes, and they would put them on the dish. And this poor postdoc in his 6.5 years in basically said, they all look the same. And so Cricket and John met me. I gave a talk over there and met me just as this postdoc was about to give up. And they sort of said, is there any reason why putting them into your devices might show us a phenotype that would otherwise be hidden when they're sitting on a dish? And I said, well, if there's differences, for example, in contractility, what you would see on a dish is that they all twitch, but you can't measure how much power they can generate on that surface. But in our devices, you can actually measure it. So maybe if there's a subtle difference, then it's something that you would be able to see. So here's the first, oh, that's odd. Anyway, here's the first set of patients that we generated cardiomyocytes from the iPS cells. The top is a wild type cell line, and the bottom one is from a patient that had dilated cardiomyopathy. And what you can see here, if I click on, you can sort of see this movie. What we observed is that this patient that had a mutation in titin had abnormal contractility in the cardiomyocytes. And what you cannot see, because all of the graphics are weird, is that we did this across multiple patients, all of whom had different mutations in titin, all of whom then exhibited defects in contractility in our assay. So we submitted this to Science. They were super excited, and they said, how do we know that this is really a titin mutation? Because these are all different patients. Yeah, you have a mutation in titin, but there could be other mutations that are involved. Is there a secondary mutation, et cetera, et cetera? So we waited 1.5 years. And during that 1.5 years, CRISPR came along, and we said, oh, here's a way we can do this. And so we took a wild type patient's cells and then generated CRISPR mutations, knockout mutations, et cetera, within titin. And we showed across 4 different cell lines with different mutations in titin that we phenocopied this response. So this let us basically show that this mutation is really causative in the context of contractility defects. These patients all have dilated cardiomyopathies. We all know that part of that is that they have reductions in contractility. So then what we've been doing with this system is now studying mechanistically what can we do to these IPS lines to rescue contractility within them, and ask, are there ways that we could translate that in the context of these patients? We've now gone on over the last 10 years, really looking at just systematically across many different mutations in various forms of cardiomyopathies. And what we're seeing is that in all of the dilated mutations, we see alterations or aberrant contractility in terms of reduced contractility. And in all of the hypertrophic cardiomyopathies, we're seeing increased contractility in those mutations. And so now we're trying to understand how those are related. Now, along the line, one of the things that we got interested in is, can we do more than just this, right? We're sort of doing 1 mutation every year with 1 graduate student or 1 postdoc. It's pretty slow. And so what we've been thinking about is, can we bring in more engineers into this project and think about how do we take this technology that I had invented in my lab, which is really not that easy to use, but actually make it more translatable. And so we ended up writing for this NSF Engineering Research Center, where we were able to pull in 20 additional investigators into this project across 7 different institutions, across engineering and medicine, et cetera, to ask, are there ways in which we can engineer these types of tissues for other purposes? And so along those lines, what we've been doing is basically taking these different technologies and sort of improving upon them. And what I want to show you, just for a couple of illustrations, is just some things that are going on. So Gordana Vaniek-Novakovic, who's a bioengineer down at Columbia, basically took this technology and sort of said, hey, when these tissues contract, they work kind of well, but they sort of differentiate to the level of sort of like a 2nd trimester fetal cardiac tissue. So it's not quite like what happens in the adult. It's not a really great model. What's missing? And she thought, maybe what happens is that as these cardiomyocytes continue to improve in terms of heart contractility, what's driving that is simply mechanical and electrical stimulation. And so what she started doing with our tissues is simply stimulating them with progressive sort of increasing stimulation in terms of rate of contractility. And what she found when she did that is she dramatically increased the maturation state of these cells. So now these cells express many of the phenotypic markers of a young adult, and the tissues become much more contractile. So this technology, again, is something that has continued to improve as a result of that. How much time do I have? 1 minute. Okay, so I'm going to skip this other piece, which is we collaborated with another group that is doing 3D printing, and basically taking this idea of, instead of making sort of a muscle strip, making a muscle chamber. And this allows us actually to create valves and all of this stuff, but I'm not going to go into the details of that. All I can say is that what we've been able to do is produce basically a heart chamber that can reproduce the PV loop of a normal heart. And now we can use this to study the effects of afterload and preload within the system. One other person over at Harvard, Jennifer Lewis, who's an expert in 3D printing, looked at our technology and said, hey, I think I can make a whole cardiac patch, multi-centimeter scale patch. Our little tissues are sort of on the order of 1 millimeter in size. What she did was she made a system where she could make millions of these little tissues and then suspend them into this giant 3D printer where the bore of the 3D printer is quite large, and then feed these guys into the 3D printer as if they are the ink themselves, and then would print them out. And you can sort of see here, just depending on how you move the printer head, you can sort of align these little microtissues into a little patch. And then what happens over time is that those cells are still remodeling. They will connect with each other to form a very large patch. And this is something that we're now using to sort of see if we can build patches for therapeutic use. Again, just to kind of give you an illustration of that. And what I'm going to end with is just saying that in addition to building heart tissue itself, the muscle, we also want to have to build vasculature as we scale up. And so that's a whole nother project that we're working on. But I don't want to go into the details of that. The main thing I want to just sort of say is that the purpose of these things is that we really want to build different types of tissues and different tissue organizations because we believe that cells alone are not really that useful. It's only when they're architected into tissue-like structures that you're able to sort of rebuild the functionalities of various types of organs, lung, kidney, et cetera. And that there's a lot of technologies that are needed to kind of make that happen. But the goal is not simply to sort of make these to replace organs, but the whole science of how you do this is something that I think is still something that needs to be built. And it has, as Daryl said, a translational mission in its underpinnings, but underneath that are a lot of basic science questions that need to be asked about how we do this. So with that, all I want to say is weirdo geometries in my talk, but that we think that you need sort of a combination of basic biology, medicine, and engineering to be working together to kind of make these kinds of efforts possible. So with that, I just want to thank you for your attention. And obviously, none of this work is not possible without great postdocs and graduate students. Thank you.
Hey, that was really interesting. Thank you. Just over here.
Okay.
So it kind of seems surprising that you get such a macroscopic defect from what are presumably point mutations in a molecule that's the size of titin. So do you have any kind of sense for titin's this huge long structural protein?
Mm-hmm.
What is it about those mutations that leads to the phenotype that you see?
Yeah, we don't know the answer for certain, but the way that the field is sort of thinking about this is that titin is sort of like the ruler for the sarcomeres themselves, right? So titin is embedded inside that MZ connection. And so if you have, these are all sort of heterozygote mutations, right? And so if you have one of those alleles missing, or reduced in some form or another, then you end up basically with a haploinsufficiency. So you have less titin within the system. And what we think is happening is that because there's less titin, they're not able to form as many sarcomeres per cardiomyocyte as you might normally form. And then as a result, that sort of triggers this whole downstream effect of reduced contractility and under load that leads to differences in remodeling that ultimately lead to that dilation. So is that?
This is kind of a naive question, but so what would it take to get from one of your cardiac patches into real world use so that humans could actually have holes patched?
Yeah, that's going to be many, many years away. The way I see it is that the first uses are like the way we're using them as simply as culture models to study mechanisms that ultimately maybe could be used to develop a new drug. So organ on chip type applications. But ultimately, obviously, if we can make these tissues with enough fidelity that they could actually get integrated into heart tissue, then there's a lot of settings where one thinks that perhaps increasing cardiac muscle mass would be useful. And in that setting, I think that could be used. But there are a lot of issues. So for example, right now there are 2 clinical trials going on with iPS cardiomyocytes. There's one that's led by Chuck Murray out in the West Coast. They take iPS cardiomyocytes, inject billions of them into the heart. And what they see in their non-human primate studies is that there's a subset of those that lead to arrhythmias because these less mature cardiomyocytes are now integrating in, leading to abnormal conduction issues. So they've identified some of the ion channels in the iPS lines that are doing that, and they're trying to make now modified cardiomyocytes for that purpose. The other group is in Germany. Wolfram Zimmermann has now been in patients. So he has an approved clinical trial going right now where they make tissues like the way we do, and they take the tissues to sort of assemble a patch, and then they place the patch over a region of hypokinesis to get improved cardiac contractility in that space. The one patient where they've reported on so far, safety, no issues, and they think there's some improvement in ejection fraction, and the work that he did with baboons showed efficacy. So it's moving, but it's very slow.
Very nice work. Thank you for sharing. Could you please comment a bit more about the importance of the matrix? You mentioned collagen, and if you mix collagens with fibronectin, and there have been other artificial ones such as silk, how would it impact your system? I sort of know what you would say, but I think it'd be nice to discuss it.
The matrix matters a lot. And it matters both in our in vitro models. So different types of matrices have different extents of remodelability, different extents of cross-linking. And what we see is that there are certain types of starting conditions that lead to sort of a fibrotic-like phenotype. Fibroblasts within or any stromal cells within the system start to get activated, and you produce more and more collagen, models of sort of fibrotic activity. And then there's others that don't do that. And then one step further, we've seen that certain types of starting points lead to certain matrices that once you implant them are either pro-angiogenic, allow more vascularization and engraftment, but others that are more fibrotic and lead to inflammation. So I think there's a lot of steps that need to be figured out. But yes, the matrix matters a lot.